Good morning, everyone. I am Mr. Harsh Purohit, Assistant Professor in the Microbiology Department at the School of Science, RK University. I hold an MSc in Microbiology and DMLT, and I'm currently pursuing a PhD. Today, I'm excited to guide you through the fascinating topic of equipments used in histopathology. Let's dive right in. Histopathology is the study of diseased tissues under a microscope. It helps us understand the structure and abnormalities of tissues, aiding in the diagnosis of various diseases. The tools used in histopathology labs are like the essential instruments in a detective's kit, helping us uncover the mysteries of tissue samples with precision and accuracy. These tools play a vital role in preparing, processing, and analyzing tissues effectively.
Here are the key equipment and concepts that will be covered. What is histopathology? Histopathology is the study of tissues to understand their structure and diagnose diseases. Biopsy, microscope, microtome, paraffin bath, slide warmer, knives, honing and stropping equipment, embedding cassettes, storage cabinet, automatic tissue processors. What is histopathology? Histopathology is the microscopic study of tissues to detect abnormalities, diagnose diseases, and understand their progression. By examining tissue samples at the cellular level, histopathologists can identify structural changes caused by diseases like cancer, inflammation, or infection. The process involves collecting, processing, staining, and analyzing tissue sections under a microscope. Imagine histopathology as solving a puzzle. Each tissue sample provides small clues, and by studying it in detail, the pathologist uncovers the bigger picture of what is happening inside the body. What is a biopsy? A biopsy is the procedure of extracting a small piece of tissue from a living organism to study it under the microscope. This tissue sample is usually taken from areas where abnormalities are suspected, such as a lump or lesion. For example, if a patient has a suspicious lump, a doctor might perform a biopsy to determine whether it's cancerous. In histopathology, biopsies are processed to prepare thin sections that can be examined microscopically for abnormalities. What is a microscope? A microscope is an optical device used to magnify and visualize tiny structures like cells in a tissue sample. In histopathology, the microscope is the final and most important tool for diagnosing diseases. How it works: light passes through a thin tissue section, and the microscope magnifies the image so the pathologist can study the cellular details. Purpose: microscopes allow histopathologists to identify abnormal cell shapes, structures, or patterns that could indicate disease. Think of the microscope as a magnifying glass for the body, enabling us to see what the naked eye cannot. What is a microtome? A microtome is a specialized instrument used to cut very thin sections of tissue, typically 4 to 5 micrometers thick, for microscopic examination. This thinness is necessary to allow light to pass through the tissue when viewed under a microscope. How it works: the tissue is embedded in wax, and the microtome uses a sharp blade to slice it into precise, uniform sections. Analogy: imagine slicing a cucumber paper thin to make it almost transparent. The microtome does the same for tissue but with much higher precision. What is a paraffin wax bath? A paraffin wax bath is used to embed tissue samples in paraffin wax. This step provides structural support to the tissue, making it easier to cut thin sections using a microtome. How it works: the tissue is first processed to remove water and is then infiltrated with molten wax in the bath. Once the wax hardens, the tissue is ready for sectioning. Importance: without this support, delicate tissue samples would tear or crumble during cutting. What is a slide warmer? A slide warmer is used to gently heat microscope slides, ensuring that the thin tissue sections stick to the slide without wrinkling or tearing. How it works: after placing a tissue section onto a glass slide, the slide is placed on the warmer. The heat causes the wax to soften slightly, helping the tissue adhere flatly to the slide. Purpose: This step is crucial for preparing slides that can be stained and examined without distortion. What are knives, honing, and stropping equipment? Honing and stropping equipment are tools used to maintain and sharpen the blades of a microtome. A sharp blade is essential for cutting clean, precise tissue sections. Honing. This process involves grinding the blade on a sharpening stone to restore its edge. Stropping. After honing, stropping is done using a leather strap to polish the blade, removing any microscopic imperfections. Importance. A dull or damaged blade can tear the tissue instead of slicing it cleanly, compromising the quality of the slides. What are embedding cassettes? Embedding cassettes are small containers used to hold tissue samples during processing and embedding. They allow tissue to remain organized and identifiable throughout the preparation process. Purpose. The cassette ensures that the tissue is securely held while it undergoes dehydration, clearing, and wax infiltration. Color coding. Different colors of embedding cassettes are used to distinguish tissue types or cases for easy identification. Yellow, liver or renal tissue. Green, routine samples, general tissue. White, bone samples. Gray, skin samples. Pink, lymph nodes. This color coding system helps lab technicians quickly recognize the type of tissue being processed, ensuring efficiency and accuracy in handling samples. A storage cabinet in histopathology is used to securely store prepared tissue slides, paraffin blocks, and other important lab materials. These cabinets are designed to protect samples from environmental damage, such as dust, moisture, and temperature fluctuations, ensuring their integrity for future analysis. Key features of a storage cabinet often include compartments or drawers, designed to hold slides or paraffin blocks systematically. Dust and moisture protection, to maintain the quality of stored materials. Labeling options, for easy identification and organization of samples. Temperature control. In advanced models, some cabinets offer controlled environments to ensure long-term preservation of sensitive materials. A well-maintained storage cabinet is essential to keep the lab organized and ensure the longevity and usability of samples for both current and future studies. An automated tissue processor is a machine that prepares tissue samples for microscopic examination by fixing, dehydrating, clearing, and infiltrating them with paraffin. These machines are used in laboratories, especially in histology, immunology, microbiology, and biotechnology. There are two main types of automated tissue processors. Tissue transfer machines. Also known as dip and dunk machines, these processors move specimens from one container to another for processing. Fluid transfer machines. Also known as enclosed machines, these processors hold specimens in a single chamber and pump fluids in and out. Modern fluid transfer processors use raised temperatures, vacuum/pressure cycles, and effective fluid circulation to reduce processing times. Thank you for watching. Please let me know if you have any questions in discussion section.